We have some housekeeping issues. First of all, I want to welcome back our live stream viewers, those people who are looking at us on the internet, which is kind of amazing to me. We encourage you on the internet to comment on the YouTube or epilepsy.com page. So we encourage you to comment on the YouTube or epilepsy.com page. And also, you may use the hashtag 2014 pipeline on social media. Now, I have to tell you, I'm not sure I know what that means, but I'm sure that there are lots of 25-year-olds out there who know exactly what that means, okay? So the hashtag is 2014 pipeline, okay? Now, some other housekeeping things, which I'll mention later again, is that lunch, okay, is served in atrium room 3 and 4, which is one floor up, and it's easy to find. The posters are behind the bar. So if you find the bar, look behind that, you'll find the poster session. All right, we must get started, and I will begin with my good friend Jeffrey Skolnick from Intellimedix, who's going to tell us about the platform of computational in-Silico screening in the zebrafish. Remember, please, everyone, 6 slides, 6 minutes. Otherwise, lunch will be at 3 o'clock. 6 slides, 6 minutes. We have 13 presentations. Jeff, where are you?
Okay, so let me cheat. This was just the title. So let me just take you through the basic pipeline ideas that we're doing. Our idea is to try to use computational modeling to prioritize drug targets. And so basically what we do is we're going to predict the 3-dimensional structure of the putatively biologically active form of the protein of interest. In practice, this covers 86% of the human proteome. We then attempt to find small molecule ligand binding pockets that are in that protein that may or may not be present in other proteins. And basically the idea is, as copying is good, you can actually copy ligands, and I'll show you a concrete example where this apparently works. Amongst the other things we can do from the computational predictions, and these are predictions, not a substitute for experiment, is to actually predict side effects with a reasonable correlation with known side effects when you pretend you don't have them, as well as toxicity. So what you basically wind up with is, in principle, you could screen 20 million compounds if you want, but the success rates, at least for small molecule binding, and as you see it for physiological response, vary somewhere between 10 and 50% for a handful of molecules, 2 to 4, 10. So you don't actually, even though we have a high throughput platform, you don't have to screen a lot. Now, of course, an in-Silico prediction is just that. It's a molecular cartoon. It doesn't necessarily mean anything. And even if you got the binding right, it wouldn't necessarily mean that you had the physiological function. So what we do is we use both zebrafish and iPSCs to confirm the activity. We then, if this were to work in a fish, and a fish is not a person, we do a mouse is not a person. And since we're interested in repurposing FDA-approved drugs, we can have a clinical trial. Now, there's a couple of ways you can do it. You can either go through the fish and the animal models, or if the toxicity profiles are sufficiently benign and the contemporary treatments are sufficiently inadequate, you can actually jump to the clinical trials. And I'll show you some preliminary results which are very promising. So this is the predicted 3-dimensional structure of the SCN1A ion channel, which is implicated in Dravet syndrome, as well as a bunch of other neurological epilepsies. And if you want to look at new modes of action, what you don't want to do is necessarily look at the binding. This is actually over here. This is the ion channel. There's 4 voltage sensing domains. The zebrafish mutant is actually in one of these voltage sensing domains buried in the core, so it's actually a weak seizure phenotype because of that. One of the things we found, interestingly enough, is there's actually a whole bunch of novel ligand binding sites, one of which actually is predicted to bind progesterone. And this was stolen from another molecule, mineralocorticoid receptor. And that's very nice. It turns out that the number of small molecule ligand binding sites is about 500. And so you can copy and paste and find interesting potential leads quite easily. That's very nice. So what we do is we look in a zebrafish. And again, I don't have the time with 6 slides in 6 minutes. We looked at the response to light. which is basically you introduce pulses, you introduce effectively a seizure in the fish. There's a series of pulses introduced. The response in the darkness to the light is down at 1. And what you see is actually, you can look at the unnormalized. You actually have to normalize for the solvent response. So in the case of progesterone at 45 minutes and 2 hours, it has a statistically significant response with a p-value of 4 × 10 to the -4th. Another molecule which came up of the two that we tested that were novel, that are putatively binding to this binding site, is mifepristone. You would have never guessed this beast. This is this fellow down here. It is also reducing the response of the light to the fish. So insofar as you can, and if you actually look at the response of known AEDs to models of Dravet and actually Dravet patients, this is consistent with the response to the other drugs. It's a little bit better than some and a little bit worse than others, but it's certainly in the ballpark, and it's a novel mode of action. This is just showing you, amongst the other things, you can actually look at calcium imaging. These are chemically induced seizures. And one of the things which should become dramatically apparent is not all seizures are created equal. The 4AP seizures are more like localized thunderstorms. The bicuculin-induced seizures are actually kind of like a global thunderstorm across the sky. And so what this suggests is, and this is one of the ways we're assessing various types of drugs, is you want to use different types of seizure patterns, and different mutations will introduce different kinds of seizures, and therefore different types of drugs may be more appropriate. And this is part of our pipeline. I pray that this thing stops and lets me go ahead. Another thing we did, just kind of showing you some very preliminary results, is we looked at chronic fatigue syndrome, which it turns out that this whole nest of diseases are not unrelated. We did entrap-1, which is a mitochondrial chaperone. A key mutation was involved in the ATPase binding site. We predicted a novel drug, which is completely out of the, with very small benign side effects. They've been actually feeding it to 2 patients and are supposed to continue the trials. And quote, the patients are up and about and energetic. Now, this is the statistics at 2 and very anecdotal but preliminary. But this shows you that by looking at FDA-approved drugs whose toxicity profiles are benign, you can actually skip to the finish line pretty quickly because at least you know it's not going to kill the patient. Whether it's actually going to do anything is a separate question. In this case, there are suggestions that it's going to do something, and now we're going to be engaged with a partner in a much larger clinical trial. Again, this was purely from a computational model. There was no way you would predict this at random. And so the trials and challenges for the future, we basically are, while the Diddy mutant is interesting, it's actually a weak phenotype of seizures because of where the mutation is located. We're actually building personalized zebrafish. There's various types of Dravet. If you actually look at the 3D structure, there's at least 6 different kinds of Dravet, depending on whether it occurs in the ion channel or in the voltage sensing domains, and some actually have gain of function, some a loss of function, depending upon what it does. And so you actually want to use personalized fish, or at least classes of representative fish, to test it. We're looking at calcium imaging, as I showed you. Is it a global thunderstorm or a localized perturbation? We're looking at personalized fish results with iPSCs, trying to correlate. Fish are easy to build. iPSCs are harder to build, but potentially much more powerful. What's the correlation, if any, between the various models? What can you conclude? We're testing progesterone, mifepristone, and we have a whole bunch of other drugs in mouse models as well as in single ion channels to demonstrate that the putative mode of action is what we think it is. We actually have lots of other molecules, and we can look at lots of other novel binding sites and play this game very quickly. So even though it is a computational model, and your initial response might be, well, it's very interesting, but it's a cartoon, it's a very useful predictive cartoon for prioritization. And depending upon the results of the progesterone and the mifepristone, we're actually trying to convince some MDs to actually try clinical trials. By the way, progesterone actually is used in a form of cannabidiol epilepsy. There's a lot of anecdotal evidence as to it does work in some patients. It was never tried in Dravet. There is anecdotal evidence, if you actually look back on some clinical trials, that it helps when girls go into puberty and are put on birth control pills. But the question is was this an accident? Or it may well not be. And this is a typical example of how you can actually repurpose binding sites to repurpose drugs and do this in a very accelerated and very rapid way. Time to market. So I thank you, and I'll be happy to try to answer questions in the femtosecond I have.
I need a live mic here, please. Thank you, Jeff. we have time for a question, and there are 3 microphones in the aisles. Since I don't see anyone there, Jeff, is this a small molecule effort in terms of finding a new therapy, or is this something other than a small molecule effort?
The answer to your question is both. It is a small molecule effort to try to find a new therapy. We're trying to find novel classes of drugs that don't have the toxicity and side effects of existing AEDs. So what we're looking at are novel binding pockets, which at least have a chance. We have no way of predicting this in advance, whether the side effects are benign. Well, actually, we can suggest whether the side effects will be or not. But this is more generally trying to, A, understand the etiology of the disease, so that's on my academic hat, as well as ways of treating it. And this is basically designed to be an extremely accelerated effort. It's very much a look here, not there, and then test it very quickly in animal models and try to move these FDA-approved drugs to a clinical trial as quickly as is possible. So it's a hypothesis generator.
Thank you very much, Jeff. Appreciate it very much. Great talk. Our next speaker is Dr. Cynthia Rask from Asklepios, who will talk about Galanin gene therapy.
Thank you.
It's that green button, and you have to hold it up. We have Dr. Rask's slides.
This seems to be going bad.
Are you pushing the green button?
Yes. We don't. Let's see. There we go. Okay.
Here we go.
There we go. So I'm going to talk about Galanin delivery, targeted delivery by gene therapy, actually. And this is not yet in the clinic, but it's part of a platform technology that's being developed by Asklepios BioPharmaceuticals, or AskBio, as it's often known. It's a small, privately held company with a technology that's supporting nucleic acid delivery for therapeutic development in multiple therapeutic areas. It's been around since 2003 and has, as I said, has a whole platform program, and I'll talk about some of the other programs that are actually in development currently, in clinical development. There have been studies in Duchenne's that have been published, that have been done by Jerry Mendel and others, and we have an ongoing trial right now in hemophilia B, and there was a spin-out of AskBio formed specifically for the hemophilia project, and that was recently acquired by Baxter Healthcare because of a good safety profile that's been demonstrated thus far, and some promising, what look to be promising results in the limited number of patients who have been exposed to date. There are also a number of preclinical gene transfer projects in hemophilia A, macular degeneration, and the one I'll focus on here is mesial temporal lobe epilepsy. And this has involved numerous collaborations, both academic and with other companies. So the technology is based on work that's been done primarily by Jude Samolsky and Dr. McCon at UNC, and there have been multiple proof of concept studies conducted thus far in a variety of diseases. And it's really an exciting time, I think, for gene therapy, because there has been a gene therapy that's been approved now using an adeno-associated virus. Glybera was approved in 2012, and so is actually on the market. And there have been a number of successes in ophthalmologic indications, which you've probably read about, and the safety profile appears now in over 60 trials or so that have been conducted to date, appears to be quite good. So it's a way of delivering a targeted neuroactive peptide to the brain, to specific areas, and without the systemic delivery. and it's a one-time administration and getting long-term efficacy and safety, hopefully. And it will build on other techniques. So why Galanin specifically? It is a localized constitutive factor, which there are multiple proof of principle studies in the literature that show that it does has an effect to attenuate seizure activity in various animal models. That Galanin knockout mice exhibit a significantly greater seizure sensitivity, whereas if they're overexpressed Galanin, they have a marked reduced seizure sensitivity. And this has been, there are a number of publications since about 2000. Here you can see a list of studies that have been done and what sort of models they've used. So I think the mechanism is quite well understood. It's being done in conjunction with the proof of concept studies that are have been used, built on technology that's what has been used previously in Parkinson's disease and in other CNS indications where the drugs have actually been delivered in a targeted fashion to the CNS. So the proof of concept studies in the public domain, there are multiple studies. A dose escalation study to determine the upper limit and maximum amount that's deliverable has been completed. Evaluation of worst case scenario, that is where you target the temporal lobe, but the drug actually, the protein actually goes into the ventricles, has been completed. And behavioral studies, again, to look at the possibility of what happens if the target is not reached. And some of the drug goes to a different target, an unintended target. And that's been completed. The current status is that we had what's called we sometimes call a pre-IND to get advice from the Center for Biologics group on design of the toxicology studies. There are preliminary studies in non-human primates and sheep, which have been completed. The delivery system has been done in collaboration with a Medtronic device, which has been in clinical trials. So the studies in these large animal models have already been done. However, they are quite expensive to do, and we're estimating that it will cost another $5 to $6 million to do the definitive dosing studies in rodents and non-human primates, and for toxicology and biodistribution studies to get to an IND. So that's the next step. Thank you.
Thank you very much. Any questions from the audience for Dr. Ask? This is a chance for the audience to participate. We have a couple of minutes. Steve?
I was just curious if you could comment on the preclinical efficacy results that you've observed.
Yes. Well, these have been done in knockout models where the Galanin gene is missing, and so delivery of Galanin actually reduces or eliminates seizures in those models.
Thank you.
In a dose-response fashion.
I presume that you don't have quite in hand the $6 million needed to finish these studies. So we'll call you as partially in the valley of death here.
Yeah. Yes.
Okay.
Partially.
Thank you very much.
Hopefully not completely.
Thank you very much, Dr. Rast.
Right.
Terrific. Mr. Amir Pessian had a family emergency and asked me to present his slides. I won't take any questions because I'm not competent, I don't think, to answer any questions. So this is AMPX 0079 from AurimMed Pharma from Utah, and I'm filling in for Mr. Pessian for this presentation. This drug is effective in models. We heard Chris Galanin talk about our models are predictive. All those lovely little pluses that you see in there tells you that this drug is effective, okay? And including, I don't have a pointer, but the mesial temporal lobe epilepsy model that was so well described by Steve White also has a plus. So this is one of the first successes out of that model from the anticonvulsant screening program. So this drug looks good when you look at the efficacy models, okay? It's been through a SERAP full bioprint. Now, I had to Google that to be sure I knew what it was. Some of you obviously know exactly what it is, but there was no significant activity. It is a CYP450 substrate, no P-glycoprotein inhibition, lack of cytotoxicity, lack side effects of these targets, minimum predisposition for drug-drug interactions. I'm not absolutely sure I understand how that squares with the CYP450 substrate, but we'll let that go since Amir isn't here to defend that. And there are implications for a novel mechanism of action. And which, by the way, is all we can hope for in terms of finding mechanisms of action, is it's novel. And if you want to look at that, there's a poster upstairs. The average age of the authors of that poster, of which I'm the youngest, is about 78. So this could be the oldest poster ever posted. Go by and look about, talks about mechanism of action. Okay, immediate plans are phenotypic screening, prediction of sedative side effects. He's interested in seeing if this is a sedative compound. Obviously, I hope that this is not a primary issue for him. But here is the real problem. This is where he needs to be. This is the valley of death. He needs analytical work, genetic tox, general tox, safety pharmacology. This is the $1.5 million that's the valley of death for so many of our compounds. And we don't have the resources yet. We're working on it. We don't have the resources yet to get to this, to an IND, where we at least have a hope, not a guarantee, at least a hope that Big Pharma will pick it up. So here are the development challenges. He's contacted everyone from VCs and pharmaceutical companies. I happen to know that. And they say, well, tell us what you've got when you're into phase 1. Well, it takes $2 million to get to phase 1. He's giving away equity. He's offering partnership, licensing. He's doing everything he can to try to make this drug work. So that's AurimMed's AMP X0079. And as I said, I won't take any questions because I'm not competent to answer them. So our next speaker is from BioCrea, Dr. Norbert Höfgen. I hope I'm saying that right. On GABA PAMS. Did I hit it? My umlaut was perfect, huh?
Where's the...
Oh, I have it. The green button pushes forward and hold it up.
Okay. Well, thank you very much. BioCrea is a small biotech company in Germany, and the good message of today is that the German organic chemists are back in epilepsy research. The team, the company is roughly 3 years old, but the team itself is involved in epilepsy research since more than 20 years now in different organizational forms. And we were able to discover and develop anticonvulsants which are at the market now. The one example is retigabine, and the other one is imipidine, which entered the market for veterinary use a year ago. The program I'm presenting today is on the GABA receptor and got the innovation seal earlier this year. Yeah. Though, as you all know, the GABA receptor, the GABA-A receptor, is not a new target. It's an old one. And we all know about the benzodiazepines and the efficacy of these compounds in several seizure animal models. However, we also know about the limitations. In epilepsy, we are faced with tolerance development, and then even all the other things like sedation, abuse liability, cognitive impairment, and so on. Though what we and even other groups would like to achieve is to use this old target, but by a new mode of action to make the value of this target accessible to therapy in a more unlimited way. And what we are using here are small molecules which do nothing at the benzo site, but are able to positive modulate the GABA-A receptor via total independent binding site. Though the benzo binding site is located, as shown here in red, at the alpha-gamma interface. As a consequence, the subtype selectivity of the benzos is linked to all the subtypes which contain a gamma subunit. Our drugs are sitting at the alpha-beta interface, which allows total different subtype selectivity, though it's not required to have a gamma unit in, though this should allow to even stimulate extrasynaptic GABA-A receptors, which is the case. So this allows additional therapeutic value, hopefully for instance, status epilepticus treatment, but even for other treatment-resistant areas and epilepsy. Though what we would expect normally, having the request for alpha and the beta subunit, is that these would be unselective pumps for all types of GABA receptors. Surprisingly, that's not the case, though the profiling of these compounds yielded that we see for the beta unit a certain selectivity, a preferred activation of beta-2 containing receptor subtypes, as shown here for the alpha-1 beta-2 gamma-2 version, kind of discrimination of a beta-1 containing receptor subtypes, and some activity on the beta-3 receptor subtypes. Though there are some, at least animal studies, indicating that a beta-1 containing subtype could be related to some of the side effects, especially sedation. We will test if this is true with this type of compounds. Could open up an avenue for better tolerability. The question is what clinical picture you could expect from this type of subtype selectivity. And going back roughly 20 years now from now, we found some clinical validation already for this type of selectivity, which is linked to loraclizole, an old drug which was in clinical development in the 90s with successful proof of concept studies. And this drug already at this time was profiled to have no tolerance development in clinical studies up to a year, no abuse liability, no withdrawal problems, was well tolerated, though maybe this was a front runner of our compounds we are progressing now. Also, it had been stopped later on due to developability issues. Though what we aiming for is to provide drugs for certain kinds of refractory epilepsy for status, and this in combination with avoidance of the limitations we know from the benzos. From the developability profile, even we feel confident that combination therapy is an excellent field for this kind of drugs. To give you a flavor on the in vivo situation, again only with yes instead of a plus, what we could expect, as known from the benzos or other GABAergic drugs, we have a broad field of activities in seizure models for generalized and partial epilepsy, but also for refractory epilepsy in the well-known 6 Hz model. The remarkable thing for the 6 Hz model is that using the different versions, 32 milliamps or 44 milliamps, we have no dose shift. The active doses are comparable in both versions, which is not very common for other anti-epileptic drugs. And with an example on the right-hand side, you even can see that the tolerability is okay in this relatively hard model. This is compared with Rotorod experiment, and It is not at the end, so in this case, we initially tested up to 100 mg per kg, where we did not see any sign of side effects on the rotarod. And this is in line with even the rotarod experiments from the screening models, where we have, even in a mouse, a TI of at least 10-fold, which is a good indicator. The key question to us was, due to the mechanism and the receptor the benzos are using, what about tolerance development? What we established is a relatively short investigation in the MS threshold model, where we used a 5 days, twice daily dosing scheme. And already after this short period of time, we could detect loss of efficacy of diazepam as a standard diazepam of about 30% between day 1 and day 5. Sorry. And compared to this, the drugs we are testing had no drop in efficacy in the same time frame. So this is an experiment at least demonstrating the differentiation between the benzos in terms of tolerance development, providing hope that we were able to overcome this problem with this new kind of drugs. Though in the summary is, on one hand, the mechanistic difference to the benzos and the opportunities for the broader therapeutic opportunities in the areas where the benzos are active, but even in additional areas. And this in combination with excellent developability. Though we are even taking care for, in the optimization loops we were running for an ideal PK profile, for instance, for CYP inhibition profile, which is clean, to be even prepared for combination experiments and to avoid drug-drug interaction problems here. And especially the combination of the lack of tolerance and the activity, the extrasynaptic subtypes of the receptor, to us makes sense to test the compounds, what we not did so far in status models, to find out the therapeutic value, especially in this direction. And as all small companies, we are looking for partnerships. Then to keep speed for the further development would need good collaborations here. Thank you.
Thank you very much, Dr. Fiffy. Dr. Rogowski has a question.
It's very interesting that this compound has a low tolerance liability, and I'm wondering how you explain that. Is it possible that it could be, for example, a partial agonist at the GABA-A receptor? Because it seems like partial agonists may have less abuse, less tolerance and issues.
Yeah, according to our data, it's not a partial agonist.
It's not, okay.
I know all the studies from the benzo time and the search for partial benzo agonists to avoid a little bit the side effect profile. This is a totally different story.
Thank you very much.
So at the moment, you don't have an explanation for the difference in tolerance between traditional
the discussions for the reasons for the tolerance of the benzos are linked to two things, I guess. One is the special binding site got restructuring, that this could have an impact on the tolerance development, and especially for the status that even the extrasynaptic receptors are not activated by the benzos, but are still alive in this situation. And only if you could activate this type, you would have a chance to have efficacy in the status. But this is hypothesis.
Mike, we have to move on, but
I have another question. Quick question.
How can I say no to Jackie French? Make it quick.
It's real quick. So from a clinical trial perspective, what do you expect to happen in the setting of benzodiazepine use? So if patients in a study of this compound were to use intermittent benzodiazepines or chronic benzodiazepines, what do you think would happen?
Are you concerned about an interaction between benzos and the GABA PAMs?
What we see is a little bit of an allosteric modulation of the binding efficacy of the benzos in the due to this kind of molecule. So the binding to this new binding site is even modulating the binding affinity of the benzos at the benzo site. It is not much, but it's a little bit. So the binding affinity is increased a little bit for the benzos. What this means at the end in the clinic is not clear, if this is a reduction of the benzo dose or if it is a risk. Needs to.
I greatly regret that I'm going to have to stop this, and my apologies.
It's so quick. How did you determine the MTD for the rotarod test? The maximum tolerated dose?
This is via rotarod.
But you said there were no effects.
No.
So how do you know
in the direct comparison of the 6 Hz 44 milliamp versus rotarod, we tested up to 100. There was nothing. In the screening approach, you are going higher, 300 or so, and then the first signs are coming.
This is a fascinating project. Our next, it's the green button, yeah, and hold it up.
Yeah, good. Good morning, everybody, and thanks, Roger, for the invitation and for the opportunity actually to be here and to tell you about our research in epilepsy at Fluorinov. Fluorinov is a small biotech company in Toronto, and we have been actually blessed to be here today to tell you about what we have in epilepsy and our latest results. Today's talk is about one of our lead molecule, FV082, as a safer orally active broad-spectrum anti-epileptic drug candidate. FV082 is a small molecule generated from a highly focused medicinal chemistry effort in collaboration with NIH ASP program, of which today I would like to take this opportunity actually to thank the ASP program, with whom we have been working closely actually for the last 3 years, and you will see some of the outcome of this effort. The background and rationale, powerful and effective strategy to control seizures, prospect freedom from seizures and adverse side effect are major challenges, as we all know. Limited number, unfortunately, of AEDs, such as Dipacot, have broad-spectrum anti-epileptic properties, and many patients remain refractory. It's about 30%. All of us, we know those statements. So the identification of a broad-spectrum AED with indication beyond epilepsy, such as pain, for example, as Dr. Galanin mentioned this morning, like pain, depression, and others are huge. So Fluorinov Pharma have developed proprietary technology to effectively synthesize novel fluorinated small molecule AEDs with significant clinical potential. And really our objective is to develop novel orally active small molecules with broad spectrum anti-epileptic with superior safety and tolerability profile. So the safety is really our driving force in this effort. And we have been using, as I said, the ASP program fully. We have been taking full advantage of this program and working very closely with the group at NIH. And to de-risk the strategy, what we have done actually, instead of working on a single class of molecules, we started actually with 3 chemotypes, structurally different, and we made about 45 compounds. So we used the decision tree from NIH, but we incorporated some key features for drug development, such as safety, DMPK, and of course, other animal models. So we took full advantage, as I said, in the right-hand box, the animal models actually we used at NIH. And again, it has been a pleasure. Somebody from Canada using this service fully, it has been a joy. And I can tell you, last month actually I got an invitation from Ontario government to effectively to tell them about this program. So they are looking for actually to implement this program in Ontario and Canada as a whole. So FV082 displays a broad spectrum efficacy profile and well tolerated in multiple AED animal models. And just what I did here, I selected some animal models actually And just to compare them to Depakote and Keppra, and in terms of safety and broad spectrum, Depakote is a broad spectrum AED, and Keppra, it seems, is one of the safest drugs AED in the market so far. So if you look at actually, if you focus on the therapeutic index of the three molecules, apparently FV082 is quite tolerable. And we got really an MTD of 082 in animals, mouse, rat, and in dog. In mouse and rat, it was over a gram, and we dosed animals with a gram of drug, and it seems that the animals were happy with the drug. So it was really a nice surprise for us. In addition to animal models actually we generated with this molecule. So FV082 exhibits a superior preclinical safety profile when compared to the two, I would call them gold standard drugs. One of the models actually I put here is the amygdala kindling in a rat, and this model actually was run at University of Toronto, and the drug works beautifully with a dose response, which was actually between 30 and 100 mg per kg, and it seems, as you can see from this slide, FV082 has worked in this model, and as all know, the amygdala kindling is a model which really would predict the clinical efficacy for the treatment of complex partial seizure. The full details of all the data on 082 is described in a poster, and I would be very happy actually to discuss the data with you during the poster session. Of course, the mechanism of action. So we were blind actually when we started this program, and we didn't have a clue about the mechanism of action. So what we did actually, we selected 082 and other molecules. It's a really, and what we did, we broad spectrum, we broad profile the drug against over 120 targets, including all the ion channels, actually, not all, but the majority of, or the bulk of the ion channels, which would have activity in epilepsy and the pain. And from this panel, as you can see, we use the whole cell patch claimed assay, the flipper assay, as well as hippocampal slides, and syrup broad profiling. And the result we came up, actually, surprisingly, that the FV082 hit the sodium channel NAV 1.8 selectively compared to all other ion channels we screened. And we screened probably about over 16 ion channels from calcium, sodium, potassium, and chlorine ion channels. From the syrup profiling, FV082 hit some androgen receptor agonist radioligand, as well as the monoamine oxidase B. And we are following on those two to determine the functional activity of those two, to see how actually those two targets involve in the efficacy of FV082. Also, 082 actually went through the acute toxicity we started first with 5 days, and really it was a great surprise to see that FV082, it was well tolerated. This is 5 days in a rat, and as you can see, the drug is well tolerated, and only mild clinical signs reported. No issues in terms of liver function, enzymes, ALT, AST, and ALP, and body weight gain, rotorod, and other neurological signs similar to vehicle, and no change in organ tissues such as heart and others, and no increase in liver mass at doses of 200 and 100, pardon, at 200 mg per kg. In the basis of this data, presently actually we are running an oral daily tox in rat and dog, and I just got an email today. We are on day 10, pardon, orally dosing with FV082 200 and 400 mg, and it seems in rat, and it seems the drug is well tolerated. So we are really very pleased with the safety of this drug so far. Of course, Dr. Galanin, as he said, the value of AEDs, or the commercial value, I should say, actually, it's beyond epilepsy. And we have such as neuropathic pain, inflammation, and also cancer pain. And this is what we are doing actually in Toronto with OICR, our colleagues in cancer group. So what we did actually, we generated some data, and again, this data came from NIH, which is really, as I said, it has been a joy to receive the data like this, and great data. So the molecule was tested in 2 pain models, the mouse formalin mouse, and as you can see, the molecule works beautifully compared to gabapentin, as well as in neuropathic pain, such as the mechanical allodynia rat model. And again, compared to gabapentin, it's at least as good, even better than gabapentin in those models. In summary, if V082 is a new anti-epileptic drug, chemically unrelated to any AED known so far, with excellent safety profile, at least so far in mouse, rat, and dog. Inhibited voltage-gated sodium channel NaV1.8 and interacted with the IR and monoamine oxidase B. And the PK is just fantastic across species, from mouse, rat, and dog. The oral bioavailability it's close to 80%. The half-life across species, it's about 3 hours. No drug-drug interaction up to 500 micromolar. So it seems the safety, it's confirmed actually with drug-drug interaction as well. In animal seizures model, FV082 possesses broad spectrum profile and demonstrate efficacy, as I said, in models of neuropathic pain. Most importantly, what we have done actually in this case, in this molecule, we developed a manufacturing process chemistry, and already we scale up this drug, and we made about over 1 kilogram, and as we speak is going through NDA enabling. And of course, the AP, which is one of the most fittest actually for the industry, already we have a US patent application filed last year. In terms of management advisor, I think we have some...
I think we're going to have to call it to close, okay?
Good. And I'll be very happy to answer any questions. But we have 2 posters, and we'll be happy actually to discuss the data with you during the poster session.
Thank you very much, Dr. Selassie. I'm sorry, we won't be able to have any questions on this particular talk. I regret we were just running too late here. Our next speaker is Dr. Harold Cohn, who's going to talk about extended neuramides. In case you don't know who Dr. Cohn is, he invented lacosamide. It's a $2 billion drug, and now he has a follow-on he'd like to tell you about.
Thank you, Roger, for the kind introduction. I'd like to begin by telling you about the compound that comprise the NeuroGate technology. We call them extended neuramides, or ENA for short. And the ENAs were devised from 2 classes of neurological agents, functionalized amino acids and alpha aminoamides. My lab developed the functionalized amino acids, as Roger had mentioned, and lacosamide as the prototype. The alpha aminoamides came out of Italy, and sulfinamide and rafinamide are 2 prime examples. Sulfinamide in phase 3 trials for Parkinson's disorders, and rafinamide in clinical trials for lower back pain. Both lacosamide, sulfinamide, and rafinamide are all potent anticonvulsants as tested at the ASP. So we recognize in these 2 classes of compound they had a common structural core shown in the box, and we felt that we can concisely bring in pharmacophores from both classes of agents into a common structural motif by embracing that part in the box, and so we devised the ENAs. We've looked at more than 50 compounds in this class of compounds. We have evaluated the compound at the ASP, and also have done self-physiology studies through most of the compounds. I'm going to talk to you a little bit about 130, but we also briefly tell you about 168, but we have several other compounds that we're very much interested in.
It's a green one.
Yeah. Okay. So let me tell you about 130. So, and as we've been seeing throughout today's morning talk, we bring in data from the ASP. It's an incredible outfit for us. We've interacted with them for more years than I'm going to tell you. But nonetheless, I show you here data for 9 different seizure tests run to the ASP. You see here in the maximal electroshock test, the compound is effective. And what we, just briefly, you want to strive for low ED50, a more effective compound, and you try to get a high PI value, which is the neurotoxicity TD50 divided by the ED50. And what you see is in the maximal electroshock test, 130 is effective in both mouse and rats, and by IP and oral. We also see it as effective in the 6 hertz at 32 milliamp, which is where Keppra was found to be very effective. We see it also effective at the corneal Kindle mouse model and the hippocampal Kindle model. These are sensitive models for partial onset seizures. And the tests in rats are 2 tests that have been advanced for pharmacoresistance and epilepsy, and you see the compound very effective there as well. To give you some idea of how effective 130 is, what I've done to take the data from the previous slide where appropriate, and I converted the ED50, the milligram per kilogram, to micromole per kilogram, so we can compare different compounds on a per molecule, on a per molecule basis. So I've accounted for the molecular weights. And I've compared 130 with lacosamide, levetiracetam, lamotrigine, topiramate, carbamazepine, and phenytoin. And you see that, again, the lower the value, the better. That 130 outperformed all these compounds in all the tests shown here. We are particularly interested in the hippocampal Kindle seizure model, and you see that in 130 is more than 14 times more effective than any of the other ASCs on the list here. So the question is, how do these ENAs operate? Both cellular physiology studies and receptor binding assays indicate that the major pathway for these compounds is the sodium channel. And in the CNS and the PNS, that what governs membrane excitability is the fraction of channels that are in the closed or resting state that can be fired upon stimulation. And you can stop that, you can control neurohyperexcitability by inactivating the channel. There are 3 major pathways which you can do so: fast inactivation, slow inactivation, and a frequency use dependent that target an inactivated state. Of these 3 pathways, we actually favor the slow inactivation pathway and the frequency use dependent pathway, because slow inactivation occurs upon sustained depolarization and spiking, while frequency use occurs upon repeated excitations. And these conditions simulate what occurs in neuronal hyperexcitability, conditions that you would see in seizures and pain and the like. So on this slide here, I provide you some data on how 130 functions. On the left-hand panel, you see the slow inactivation curve for 130. I compared that with control, no drug, and also lacosamide. Lacosamide is, of course, the principal mode of action has been attributed to that compound by its ability to induce slow inactivation. The lacosamide curve, the red curve, was done at 100 micromolar. The 130 curve was done at 10 micromolar. And what you see is that the channel availability markedly decreased in the presence of 130. It turns to be a concentration-dependent process, allowing us to calculate the IC50 values for slow inactivation. The lower the number, again, the more effective. And when you do so, 130 is 100 times more effective than lacosamide in inducing slow inactivation. We have other compounds in our repertoire of compounds that go as high as 600-fold more potent. On the right-hand side of the curve, we show the frequency use inhibition curves for just 130 with no drug, 1, 2, and 6 micromolar. And again, you see, as with repeated pulses, you see more and more of the channel inactivated, suggesting that the compound actually either to target or to enhance an inactivated state upon repeated firing. And so, and it's been very important from my perspective, is to show you that that 130 is working at very low micromolar concentrations, and we believe that these are the therapeutic concentrations the drug would be effective. So we think these ENAs have 2 competitive advantages. One is they powerfully induce slow inactivation, and second is that they have a frequency use inhibition, again at therapeutic concentrations. So this summarizes a little bit about the ENAs and tells you a little bit more. First of all, the ENAs, like 130 and 168, have excellent activities in the seizure models with minimal neurotoxicity. We've examined the ENA, that a number of receptor bindings studies at targets known to adversely impact drug development, and we see no interaction. We see no hERG channel activity up to 30 micromolar and no CYP450 inhibition. In PK studies in rats, we have excellent drug-like properties with bioavailability that exceed 80%. They have low clearance rates and relatively high Tmax and T1 half values, and they're brain penetrant. We think that they have lower development risks because they're designed from for compounds that have been tested in man. And the compounds act as the functional inhibitors by targeting slow activation frequency use inhibition. And finally, they have longer duration of action, and they show activity in pharmacoresistant models. Lastly, I wanted to thank a few people here, the University of North Carolina Track System, North Carolina Biotechnology Center, the Epilepsy Foundation for Innovation Award, thank you, Roger, and the NIH for a Phase 1 grant. I, of course, very much indebted to the anticonvulsant screening program. I can't tell you how important they are to the program, and to Raj Khanna at the University of Arizona for the cell physiology studies, and you for your kind attention.
Thank you, Dr. Cohen. We have time for a question. So let me just ask the obvious question, Harold. Sure. A nice, deep-pocketed partner would be helpful. Is that correct?
Yes.
Okay, another drug trapped in the valley of death. We have another minute, so I think I will just say, and maybe you would like to elaborate on this, that we haven't given the anticonvulsant screening program, I think, enough credit. The last two speakers done a great job. But all the data that you've been seeing throughout this session has been generated by the ASP. And without the ASP, I don't think we'd be having this meeting. It certainly wouldn't be the same kind of meeting. So we're very indebted to NINDS and the ASP. You would agree with that?
Sure. There's no other organization that provide that kind of insight and service and a joy to work with, actually. So.
Terrific. Thank you very much, Dr. Cohen. Our next speaker is Dr. Tom Satula, who's going to tell us about NeuroGenomeX. It's the green button.
Right. Thank you, Roger, and I'd like to thank the program organizers for the opportunity to speak with you today about the development of 2DG as a therapy for epilepsy and traumatic brain injury. This development effort is an academic and commercial partnership involving the Departments of Neurology and Medical Physics at the University of Wisconsin, where the therapeutic properties of 2DG were discovered, and NeuroGenomeX, which is a Madison, Wisconsin-based commercial entity seeking to advance this therapeutic opportunity into human use. The work that's been done relative to the Valley of Death that Roger has highlighted earlier this morning, I think 2DG is climbing out of the valley, and it's been done through some of the agencies you've heard about, the ASP, the NCATS Bridges program that was also mentioned, WARF, the Wisconsin Alumni Research Foundation, the Department of Defense, private investment, and lastly, the Epilepsy Research Foundation, the Epilepsy Therapy Project, and EF, who have actually funded what will be, we hope, the first clinical trial use of 2DG in another year. It was funded back in 2009, and they have displayed extraordinary patience waiting for all this work to get done. So a lot of people have made this happen. I know you can't see the bottoms of the slides farther back in the room. 2DG is 2-deoxyglucose. It differs from sugar by the removal of a single oxygen atom. And with the removal of that oxygen atom, it blocks glycolytic metabolism. It's been a very well-known molecule. It's been used in PET scanning, which you see there on the lower left of the slide. That's a PET scan from a 9-year-old in status epilepticus, and you can see the labeled form of 2DG, FDG. It is concentrated at the site of where the seizures are originating. So this is a molecule and a potential drug that goes to where it needs to go. The mechanisms of 2DG are unique compared to all known anticonvulsants. And I'm going to talk to you briefly about the neuroprotective as well as the anticonvulsant mechanisms. We have data that it inhibits the release of certain subsets of synaptic vesicles, and it also has chronic effects which involve the metabolic regulation of gene expression related to neural circuit remodeling. The studies, preclinical studies for epilepsy have included ASP performed work in the 6 Hertz stimulation model, Fring's auditory audiogenic model. Other investigators have demonstrated therapeutic effects against status epilepticus induced by canic acid and pilocarpine. And our lab worked initially on the effects of 2DG in the kindling model, which is a chronic model of epilepsy progression, and it produces twofold slowing of the rate of circuit remodeling in epilepsy. This is a bar graph showing a 2DG treated animal during the course of kindling in gray with the normal rate. And you can see it takes about twice as long for the progress of the epileptogenic process to occur with olfactory bulb kindling, and similarly twofold slowing with perforant path kindling. So this pattern of effect in this model is, by definition, a disease-modifying effect against the progressive effects of repeated seizures. And in addition, as mentioned, it has the acute anticonvulsant activity in the acute models. These are unlike any other known epileptic medications at this point, and therefore a novel opportunity. So one of the really interesting things about 2DG is you see again the PET scan on the left side there of the 9-year-old girl in status epilepticus. 2DG goes to where glucose is being used in the brain. And sites of pathology, both acute and chronic, are frequently accompanied by excess energy metabolism. So glucose is needed to drive a lot of those processes. 2DG goes to where the action is. On the right-hand slide there is a rat brain in which perforant path afferents to the hippocampus are being stimulated at 1 Hertz, and you can see they're labeled 2DG going into the hippocampus. So this unique property of 2DG as an agent depends on what's known as activity-dependent neurovascular coupling. It's a very complex, tightly organized physiological process of delivery of oxygen and nutrients to neural circuits on the basis of neural activity. This is a fundamental property, and 2DG basically exploits that property therapeutically. So what are the implications of these findings for clinical application? We believe it will be useful for epileptic conditions in acute settings like status and clusters of seizures. Because it is delivered to the brain as a function of seizure and energy demand, you can give it, and we have evidence for this in rats, that if it's given after a seizure occurs, it has the same therapeutic effect relative to the next seizure as if it was given preferentially. So this may be an after-seizure medication. And it, I think, offers, for reasons you see on the 2 slides there, the scans and the glucose utilization images, potentially novel methods of delivery in combination with stimulation and device therapies that you heard about earlier this morning. So I'm going to move now to the neuroprotective aspects of 2DG and traumatic brain injury. This is work funded by the Department of Defense, and the model we have used is to induce traumatic brain injury in one of 2 different sites in the brain, a temporal and a frontally located site in the rat brain, and give 2DG at a dose of 250 milligrams per kilogram per day for 2 weeks, starting at the time of the injury. And we have used in vivo and ex vivo imaging in rodents at 1 week, 1 month, and 6 months to look at the effects of 2DG as a structural neuroprotective agent. Because of time, I'm not going to go into detail here. This is an MR scan, serial MR scans in vivo from rats that have had brain injury. The white, this is a T2 image, so white means there's less brain substance there, and you can see in these there's a tendency for the increase of the white there in the lateral ventricle volume to indicating diminished overall brain structure and size. When this is quantitated in animals treated with 2DG during that 2-week period, the rate of secondary progression of the injury is reduced. So the initial injury is not reduced by 2DG, but the progression over a period of a month of the structural damage is reduced. And that's seen statistically significantly from the 1 week to 1 month interval, and the trend is there at 1 week to 6 months as well. This is another, again, in the interest of time, not going to go into detail. The 2DG also reduces overall lesion volume looked at 6 months after injury. Using diffusion tensor imaging, which is a relatively new but now rather well introduced imaging modality to look at microstructural organization in tissue. There's also been evidence of 2DG's effects on delayed development of abnormalities in mean diffusion. This is the same paradigm as indicated on the schematic at the top of the slide. 2DG treatment reduces the progression of mean diffusion abnormalities. Finally, I'm just going to summarize this last slide quickly. These structural neuroprotective agents are accompanied by functional therapeutic effects on long-term outcome after head injury in a fear conditioning paradigm, which I won't go into detail here. Basically, 2DG reduces fear learning at 1 month after injury in rodents, and at 6 months after injury, it enhances the retention of spatial memory that would otherwise be lost. So this is a structural and functional neuroprotective effect in rodent TBI. And my last slide here summarizes where 2DG is in the pipeline. There are 2 use patents for epilepsy, an Australian patent for epilepsy, pending patents in Canada and Europe. There's an issued patent for structural and functional neuroprotection in TBI. The animal toxicology has been favorable to this point. At higher doses, 2DG produces cardiac toxicity, which is not surprising based on the reliance of the heart on glycolytic metabolism. That is reversible, and the no overt adverse event level is about twice what the doses that will likely be used for neurological effects. The GMP production through NIH Bridges program is completed. Toxicity studies initially in rodents and dogs are completed, and we're expecting to be starting the GLP tox study shortly. We're also very fortunate to have, as I said, the patient support from the Epilepsy Therapy Project and the Epilepsy Foundation, Epilepsy Research Foundation for the first clinical trial, which we hope will begin pending IND approval at the FDA with Nathan Fountain at the University of Virginia. So I'd like to thank you for your attention.
Thank you, Tom. I don't see anyone at the microphone. We do have one question.
Does the systemic glycolytic inhibition with this elevate blood ketone levels, and does it also elevate adenosine levels?
We're not aware that it has those effects systemically. The question, I think, is a good one. The way we got onto this was thinking about why the ketogenic diet works, so the interest in ketones
like the ketogenic
was
diet
part
of
of
pill,
yeah.
sort of, yeah. Okay, thanks.
Thank you very much, Tom. Our next speaker is Mark Davis from INSYS Therapeutics on synthetic CBD. It's the green button.
Thank you very much, Dr. Porter, and thank you to the organizers for giving INSYS the opportunity to come and present today on cannabidiol in epilepsy. Given that I only have 6 minutes, I promise not to read this slide word for word. As an introduction to INSYS Therapeutics, we are a commercial stage pharmaceutical company, and we have products that we are using in cancer settings. Specifically, we're using a proprietary sublingual spray technology for fentanyl. And we're using that same technology for multiple platforms and looking at other opportunities for that, using that proprietary delivery system. We also have an extensive platform in cannabinoids. We have launched a generic product for Marinol called Dronabinol SG capsules, and that's a THC-based product. And we're in the process now of preparing to submit a license application for an oral formulation of Dronabinol, which again is the THC-based product. And then the company has a commitment to a cannabidiol, specifically development project. So to get into that a little bit, anybody in the room knows that there's a lot of anecdotal evidence for the use of cannabidiol in epilepsy. There's been a recent CNN article or presentation on it. We know of families that are moving to Denver, Colorado in order to be able to get access to realm oil and other things that they really believe are going to help them with their epilepsy, especially parents taking children. And there's a lot of state initiatives in place right now for getting cannabidiol into the use in epilepsy. So INSYS has developed a pharmaceutical cannabidiol alternative for treatment of epilepsy. Just a little bit about the cannabidiol. It is produced via synthetic reaction and purification process. It's not a plant-based, it's a synthetic-based cannabidiol. It's consistently and reproducibly manufactured per GMP guidelines. It is greater than 99.5% pure. And INSYS has its own manufacturing plant in Texas, where it's an FDA and DEA approved facility for manufacturing these cannabinoids. Sorry. So there at that plant, we produce the dronabinol and the cannabidiol. And importantly, this is scalable to commercial volumes, so we believe we're going to be able to supply this for the market in good quantities. Just quickly, slide is just showing the simplicity with which we manufacture this. It's synthesized chemically. It's purified through liquid chromatography. The solvent is removed and then it's ready for formulation. And one thing I noticed I didn't have on these slides is this product will be an oral formulation, and we designed that specifically partially for getting into children with epilepsy here, using an oral formulation. So in terms of the cannabidiol specifically in epilepsy, the company is committed to moving forward with this. We're looking at adult and pediatric epilepsy indications. For adult, we're looking at the treatment refractory local or focal seizures. And in the pediatric indications, we're looking at Dravet and LGS. We are getting close to filing an IND so we can start clinical trials on this product, and we hope to get that in 2014. And we've hired a CRO to help us with the whole regulatory and development strategy. And then we've also filed for orphan status in Dravet and LGS, and we're hoping to hear soon on those two. And we're engaging our key opinion leaders, many of whom are in this room, to help us with the whole development of the protocol and treatment in these patients. And lastly, I just wanted to say that there are a number of other potential indications that we're looking at for cannabidiol. As you can well imagine, as many of you know, there's a lot of noise going on about the use of medical marijuana in a number of different indications, and we believe that we have a good alternative to address some of those, specifically in helping with cocaine addiction. There's data to show that it works in glioblastoma and in peripheral neuropathic pain. So we are working with leading universities in these and starting to get some, again, traction to get trials underway as quickly as possible. And we're looking at even additional indications beyond these. So we have an exhibit set up upstairs if you'd like to come by our booth if you have any questions on the product. And that's
Thank you very much.
Thank you.
We have some time for questions.
Can you say a word about the IP situation of cannabidiol versus U.S. versus Europe?
No. It's a good question. I don't really know the details on the IP situation.
Let me ask you a question that since you can't answer that one. Something occurs to me. Are you going to have to do phase 1 with CBD, or are you going to be able to figure out a way to get around that?
We really want to understand the pharmacokinetics of this product. There's a lot of data in the literature on the use of cannabidiol and of THC and other products like that. So we're depending on that as a guidance point, as a starting point. But we need to understand what our own pharmacokinetics are with this particular formulation in an oral solution.
Do you want to know anything about the bioavailability of CBD?
It's low, but we are finding doses, or we're going to dose to make sure that we cover for the lower oral bioavailability.
That would imply greater variability too.
It could, but we'll see that. We'll be looking for that in our clinical trials. Yeah.
Okay. Thank you very much.
Okay, thank you.
Our next speaker is Dr. Steve White, who's already spoken today. He's going to tell us about Galanin receptor 2-based therapy and neuroadjuvant. Steve?
Thank you, Roger. So I want to, we've already had a very nice introduction as to the reason behind Galanin as a potential neurotherapeutic from Dr. Ask. And, you know, this Galanin, as we heard earlier from Dr. Gallon, is one of that 100+ compounds that is a modulator of neuronal function. And what I want to do today is tell you a little bit about A10-2 is a metabolically stable blood-brain barrier permeable Galanin receptor 2 preferring neuropeptide. And A10-2 is a lead compound that has come out of a series of 100+ synthesized neuropeptides from Dr. Greg Boulai's lab. And shown here is the receptor binding affinity for both Galanin receptor 1. You can see has a Ki of 494 versus Galanin receptor 2 is a Ki of 32, giving A10-2 a 15-fold affinity for GALR2 over GALR1. The anticonvulsant profile of NAX A10-2 is shown here, and you've heard a lot about the 6 Hz seizure model. You can see that it's effective, very, very potent in this model, both the 32 milliamp and the 44 milliamp relative to levetiracetam and carbamazepine. It's also active in the audiogenic seizure mouse, the corneal kindled mouse. It is very much like levetiracetam in respect to being inactive in the mouse MES test and the mouse pentyline tetrazole seizure model. Again, what is really unique about this and really exciting about it is it's a novel target in which we really don't have any human proof of concept at this point about its overall role in seizures, and particularly therapy-resistant seizures. So the goal of neuroadjuvants really is to get this program through the IND enabling talks and get it into proof of concept studies in human patients, and then move forward from there. As I've just said, it's mechanistically novel, active in several preclinical models, metabolically stable. In serum assays, it has a half-life of about 9 hours and about 25% serum. It's inactive in the hERG assay up to 30 micromolar. No effect on plasma glucose levels at therapeutic doses. This is important because GALR1 receptor is actually on the pancreatic beta cells, beta islet cells, and GALR1 activation in the pancreas actually does elevate plasma glucose, and we've demonstrated that there's a good therapeutic index with regard to that as a possible adverse effect. It has a favorable CYP induction inhibition profile. And what I'm not going to show you today is it is also a potent analgesic in several of the rodent pain models, including formalin, sciatic nerve, chronic pain. A10-2 has a favorable pharmacokinetic profile following IV administration to mice. It has a half-life at varying doses, 4 different doses, up to 1.5 milligrams per kilo, IV of around 1.2 hours. You can see that data here. And when you look at the relationship of the pharmacokinetics, or the plasma levels, I'm sorry, to its efficacy in the 32 milliamp 6 Hertz test, you can see that there's a very nice linear relationship between plasma concentrations of A10-2 and percent of animals protected in a top graph there. And shown below in the bottom graph is the percent of animals that are impaired in the rotarod following IV administration again. So the overall therapeutic index is estimated to be about 3 in this particular test. So our preclinical development activities are to finish conducting the broad receptor screening studies, which were started last fall, and those are ongoing. Dose range toxicology studies and pharmacokinetic studies in the rat are planned for the third quarter. Our pharmacokinetic studies in non-human primate, we hope to get started in the third quarter of this year, and then having a pre-IND meeting by the first quarter of 2015. So the intellectual property of A10-2 and some of the other neuropeptides is protected by two patents, international patent application issued in May of last year, and a US application. So I'd like to acknowledge a number of different people. Greg Boulai is a professor at the University of Utah in medicinal chemistry. He was really the brains behind this project and what he's done in conceiving a way to take a normally very metabolically unstable peptide or neuropeptide such as Galanin and actually change that so that it, number one, can get absorbed, number two, it can cross the blood-brain barrier, and number three, shows activity. And a number of folks there listed in his laboratory. By way of full disclosure, Karen White there is indeed a relationship. She is my daughter, and she worked with Greg during her early on in his lab as a summer student, and neuroadjuvants, the team there, Hunter Jackson, Ted Stamley, Brian Klein, who's now moved on and would normally be giving this presentation, Cameron Metcalf, and two others. The funding for this, actually, one of our very first grants, thank you again to the Epilepsy Therapy Project, for that, the Epilepsy Research Foundation, Upstart Ventures in Salt Lake City, and a U01 translational grant from the NINDS. And with that, I'd be happy to entertain any questions.
Thank you, Steve. Mike, you have a question?
Really interesting data and great progress, Steve. How do you imagine this product is actually going to be used? It's, I guess, a parenteral product, so how would you use that?
That's an excellent question, Mike. I mean, right now we are moving towards the formulation is an IV formulation. And we have, as you saw, very good PK, linear PK following IV. Our goal is really to get through the IND enabling tox studies, to get in proof of concept with an IV formulation, to get some clinical data in an effort that, yes, we are looking for partnership and support, that there might be, you know, an interest in this and to then begin to think about other delivery routes. We do have some data, limited data following intranasal administration, so one could consider that as a potential route. We also have subcutaneous administration demonstrating efficacy. That could be a potential route of administration. One area in which we actually could consider exploring is status epilepticus, as well as cluster seizures.
Thank you very much, Steve.
Thank you, Roger.
Our next speaker is Dr. Dominic D'Agostino. Am I close? I'm sorry. The ketogenic compounds.
Yes.
That's the green button on there.
Okay. It's great to be here, and I appreciate the invitation to present today. I'd like to give a special acknowledgement or thanks to the Department of Defense and the Office of Navy Research for funding this work. And in particular, it aids the Navy SEAL divers that are at risk for CNS oxygen toxicity seizures. I'd also like to give a special thanks to the Charlie Foundation. The Charlie Foundation really inspired me to pursue this area of research in metabolic therapies. In particular, the ketogenic diet for the metabolic management of seizures. So the ketogenic diet is a high-fat, low-carbohydrate, very restricted diet that essentially generates ketones through fatty acid oxidation in the liver. Now, ketones represent a water-soluble form of energy that can cross the blood-brain barrier, and it provides an alternative fuel to aid in brain metabolism. So on the ketogenic diet, the brain shifts from a glucose-based metabolism to a fatty acid and primarily ketone-based metabolism. Ketones can change fundamentally brain energy metabolism in a way that controls seizures through mechanisms we're just starting to understand now. So what's important is that the ketogenic diet, to maintain a level of therapeutic ketosis, the carbohydrate restriction needs to deplete liver glycogen stores, and you need to maintain a suppression of insulin to drive hepatic ketogenesis. This is difficult to sustain. Our lab has developed and tested a number of ketone supplements in the form of ketone mineral salts and also synthetic esters. A single administration of a ketone ester can rapidly and sustain blood levels of ketones above and beyond what can be achieved with prolonged fasting or the ketogenic diet. We can elevate blood levels of acetoacetate, which has an important role in seizure control, and beta-hydroxybutyrate. So the elevation of ketone levels can be sustained for a single administration of a ketogenic agent in the form of a mineral salt preparation or an ester, causes a rapid ketosis in the time frame of about 30 minutes. And interestingly, it can also decrease glucose levels. And it elevates, we've been able to achieve preparations that can elevate all three ketone bodies, beta-hydroxybutyrate, acetoacetate, and through the decarboxylation of acetoacetate, breath acetone levels are elevated. So there's also, with a single administration, a sustained increase that is dose-dependent in the blood levels of ketones for over 8 hours. In a recent study, we've demonstrated that therapeutic ketosis with a ketone ester can delay central nervous system oxygen toxicity seizures. So with the single administration, we found that the latency to seizure can be delayed up to 575%. So this is above and beyond what we've been able to achieve, or anyone's been able to achieve, with any anti-epileptic drug or any experimental anticonvulsant drug. We currently, we have ongoing testing in at least 4 different seizure models, and we're also soon to start a glucose transporter type 1 deficiency syndrome mouse model. So the original application for this technology was for special ops diving. In particular, the Navy SEALs, they use a closed circuit rebreather, and a limitation of their dive operations is CNS oxygen toxicity. So therapeutic ketosis, we found, increases physiological resistance under these extreme environments. Hyperoxia, and we're also looking at hypoxia. And work funded by DARPA and work in our laboratory has shown that in animal models and in humans, there's a significant increase in physical performance and cognitive performance. I don't have time to show you the data today, but these experiments are ongoing. So the main thrust of our research now is looking at the anti-seizure mechanism of these exogenous ketones. And I can't go through all of it, but what we have shown conclusively is that there's Big elevation in serum and hippocampal ketone levels, including beta-hydroxybutyrate, acetoacetate, and acetone. There's a large 5 to 10-fold increase in adenosine levels, which has been shown with the ketogenic diet. There's a large increase in the GABA to glutamate ratio, and there's also a 2 to 5-fold increase in all the TCA cycle intermediates, consistent with an increase in brain energy metabolism. We've also seen consistently that elevating ketones has an anti-glycolytic effect, and there's a decrease in blood glucose, in insulin. There's a suppression of oxygen-free radicals, and a general suppression in oxidative stress through a direct and also through an epigenetic mechanism. So in summary, we've shown that ketone supplementation can produce fasting levels of ketosis without the dietary restriction of carbohydrates or calories. Therapeutic ketosis with a ketone supplementation delays seizures, tonic-clonic seizures from oxygen toxicity, and a number of other seizure models we're studying now. There are multiple, with metabolism, you have multiple anti-seizure mechanisms that are contributing to this, and we're starting to dissect the mechanisms now. There's a pending FDA, generally recognized as safe approval. We're doing all the studies to meet the GRAS application for this approval, and we hope to start a phase 1 pilot study within a year. So thank you for your attention.
Thank you very much. Dr. French.
Very, very interesting and promising looking. I did see on one of your slides that glucose levels go down.
Yeah.
Is that potentially a safety issue, or can you speak to that?
I should have mentioned that when you take these products, you can actually lower blood glucose to 1 or 2 millimolar and not have any signs of hypoglycemia. So the therapeutic implications of that are tremendous. So insulin shock, there's a variety of things. So ketones replace brain energy metabolism in the face of hypoglycemia. And by lowering blood glucose, that could also be another mechanism that it's having an anti-seizure effect. So it's pretty profound. Like above, we do a lot of work with metformin, and it's a lot more potent at lowering glucose than, for example, metformin. So hepatic glucose output is reduced, we think, and it's also has ketones can enhance insulin sensitivity. So there's an effect there. So we're looking at diabetes models and stuff too, and it's very effective for controlling glucose.
Last question.
Thank you. My name is Adrienne, I'm Bay Area epilepsy health coach. This is fascinating. The question that runs through my mind is, since it's not done in the context of a dietary shift, what ideas do you have? I think about red, yellow, blue packets and kind of how the body gets geared up for sugar when it doesn't really receive sugar. Any ideas about what's going on outside of the dietary changes?
Very good question. My original application was this would enhance the ketogenic diet, and I really thought that carbohydrate restriction was needed to produce the physiological and neurological effects. Now, the ketogenic, most of my research is actually still with the ketogenic diet, and this is more for a military application, but what I think personally is that a large amount of this substance really needs to be administered for 1 gram per kilogram range to really get that starvation level ketosis. I see it as a way to augment the ketogenic diet. So we know a 4-to-1 ratio ketogenic diet is very restrictive, but an Atkins-like diet, a 1-to-1 ratio ketogenic diet is very sustainable, and it has therapeutic effects. And a ketone supplement 2 or 3 times a day would boost ketone levels above and beyond the classical ketogenic diet. So I see it fitting in as a way to augment a less restrictive ketogenic diet.
Thank you very much. Our next speaker is D. Scott Edwards, Vice President, General Manager of Cyflor Life Sciences.
Had competition for the podium right there. So.
No problem. It's the green button.
Yeah, thank you so much. So I'm going to spend the next 6 minutes and literally 6 slides telling you about a compound that we have in preclinical development that is a potent and selective potassium channel KCNQ2/3 activator as a potential anti-epileptic drug. A little bit about Cyflor. We're a drug discovery company. We're a chemistry company based in Cambridge, Massachusetts. So unlike many other companies, we do all our chemistry in-house and we outsource all our biology, either through CROs or through academic collaborations, which I shall describe a few of those. And we too are incredibly indebted to the NINDS ASP program, and I like to reiterate the thanks that they've been getting, well deserved. When we started in a couple of years ago, we looked for targets where we felt that using our chemical insight and some very strategic incorporation of fluorine into those small molecules, we could make improved compounds in terms of their pharmacologic properties. The first one we picked, which was an interesting choice given the time frame, was in fact the potassium channel activator retigabine. And those of you probably know that choosing that 2 years ago led to a little bit of consternation, because as soon as we started working on it, then a lot more started being known about the toxicity and latent clinical toxicity of retigabine. When we started, we were really focused on looking at what was in the package label at the time, which were the potential for urinary retention, QT prolongation, and just thought if we could design a molecule based upon what's known about the SAR, how it binds to the potassium channel as a positive allosteric modulator, we might be able to modulate the molecule and improve those properties. And then all the significant boxed warning and other restrictions on use came in. However, we did go ahead and make a small set of compounds shown on the lower right of this first slide, where you see just making 4 compounds. We actually identified out of that SF034 or SF34, which has some very interesting in vitro properties. If you look at it in terms of its activity as a potassium channel, it's about 10 times more potent in terms of KCNQ2/3. It's probably best just to look at the table on the right-hand side, upper right, where I've kind of captured the take-home message from all the graphs on the upper left, which is comparing SF34 to retigabine and contemporaneously in these models. The potency is about 10 times higher. The selectivity for 2,3 versus potassium channel 4 is improved, and the selectivity for 2,3 versus ERG is H-ERG is significantly improved as well. The reason for 4, as opposed to looking at other subtypes of this potassium channel, that work is ongoing, is because of the suggestion, at least in the literature based on rat data, that some of the urinary retention issues with retigabine could be linked to that particular potassium channel. So having better selectivity for 2,3 versus 4 could be an advantage. Data at the bottom is an example of the type of data we generate through our academic collaborations. We're working with the group at University of Connecticut, Professor Tsingunis, looking at electrophysiology and pyramidal neurons. The 2 lower left graphs in black are from control mice. The 2 lower right graphs with the red curves are from a potassium channel KCNQ2 conditional knockout mouse model, showing that you do see an effect of both ours as our compound and retigabine in the control, and much, much less of an effect when you knock out the channel as expected. As I mentioned, the NINDS has been invaluable to us in terms of testing our compound in vivo. We've been through the qualitative and probably a good part of the intermediate stage testing, and you can see we continue to show very good efficacy, ED50s that are at least as good, or in many cases somewhat lower, meaning superior to retigabine. In some cases, the protective index is significantly better, both in mice and in rats. These studies were actually done contemporaneously with retigabine that we synthesized and supplied to the NINDS. It's not comparing to the historical data. The compound has advanced in our labs with some additional preclinical testing, the types of things that you would normally do to advance something to an IND, looking at some preliminary ADME and PK, looking at clearance. In general, it's very comparable in rodents to retigabine. Looking at in vitro ADME and protein binding for mouse and human, comparing it to retigabine. In general, it looks very much the same. No significant CYP inhibition for 3 of the major CYP isozymes of concern. The additional testing that's been done, genotoxicity, Ames test negative, synthesis is readily scalable, formulation is readily attained. The intellectual property has been published as a PCT within the last 6 months or so. So the intellectual property is covered. And then last and not least is sometimes when you think about the chemistry that you've done and the types of changes you've made to a molecule and the way in which we modified a particular ring on the right-hand ring on retigabine by incorporating a fluorine, we knew that that would also potentially affect the electronics of that ring. And we had a chemical theory as to why retigabine itself undergoes this discoloration, even as an API and certainly in solution. And very simple, in essence, a high school experiment where you compare our compound versus retigabine in solution in the presence of UV light, and you can see that, in fact, that theory at least was borne out in terms of preventing this blue discoloration. Now, the relevance of that to the clinically manifest blue dermal discoloration is still an open question, but it's still an example of being able to at least find an additional potential advantage for our molecule versus the approved drug. Like I said, 6 slides. So answering the questions that were presented to us in terms of where we are in our preclinical development and future plans, we believe that making a compound with a very specific fluorine substitution pattern results in increased potency and selectivity for KCNQ2-3. We also have this finding that's much less susceptible to blue discoloration. It is proprietary. It's a new chemical entity. We have an expanding preclinical data set thanks to the NINDS, and would be approximately 12 months from being able to submit an IND. For us, obviously, the key development challenge is really understanding the implications of the findings with retigabine in terms of the toxicity that have resulted to the recent severe label restrictions. If that's mechanism-based, which we don't believe it is, but we don't know, we don't have insight into that, that's a significant bar. If it's not, then maybe improved potassium channel activators have potential utility. And therefore, we're exploring what's the best path in terms of development or partnering. And that's gated not only by getting more visibility into the toxicity of retigabine, but also by some very interesting biology that's come out in some other areas, other therapeutic areas, where having a potassium channel activator could play a role. There's recent data where retigabine is being used being planned to being used by Mass General as a treatment in ALS patients, basically translating from in vitro stem cell motor neuron testing directly to the clinic. There's a collaboration we have with a group at the University of Pittsburgh that's exploring retigabine in animal models of tinnitus, because the hyperactivity of the acoustic nerve actually correlates with the development of tinnitus. And there's also obviously an increased interest in potassium channel activators, particularly around the very rare encephalopathies that have been actually termed within the last 2 years, KCNQ2 encephalopathy. So while there's considerable questions around retigabine and the mechanism of this toxicity, there's still, we believe, some potential use of developing improved KCNQ2 channel activators, and we believe SF0034 is potentially one of those. Thank you very much.
Thank you. So I spent 15 years of my life developing retigabine only to see it stub its toe. The efforts to manipulate felbamate into a non-toxic drug and come out with a new drug failed because Big Pharma isn't really willing to take the chance. I agree that you have a great hypothesis about the coloring and you have some great data, but you admit that this is hypothetical when it comes to the human, and we're really not sure of the mechanism. Do you think Big Pharma is going to be willing to take the multi-million-dollar chance of putting this drug in the clinic, especially when it takes so darn long to find the discoloration?
Obviously, it's a very good question, and part of that is gated not only by the amount of investment and time it would take, but also potentially by some of these other indications that may actually pull it forward because there's more interest in some of those indications. So while originally we set out to look at it as an AED, it could be that we're able to advance it because of interest in some of these other earlier stage but more pharma interest indications.
Last question.
Perform a metabolite ID, and how does this compare to retigabine?
It's ongoing. Thank you.
Okay, thank you very much. Our last speaker of this morning is Dr. Jeffrey Jonas from Sage Therapeutics, following which I will have some, well, since he's taking his time getting up here, let me give you the housekeeping announcements. I won't penalize you for the time that I'm taking here. Immediately after his talk, the lunch is served in the atrium 3 and 4, which is one level up, okay? And also, please visit our posters and exhibitors, which is behind the bar, okay? So that's easy to find. Even for me, it's easy to find them. We will begin very promptly at 2:10 with Dr. Friedman organizing the first part of the afternoon session. So lunch in Atrium 3 and 4, please visit our exhibitors. They're supporting us, and we would like to have you come by and say hello to our exhibitors and our posters. So without further ado, Dr. Jonas.
Well, thank you all, and thank you for giving me the coveted slot before lunch. So I want to thank you all for being here today, and if my stomach starts grumbling, then I'll be at Atrium 3 sooner. I want to personally acknowledge all of my SAGE coworkers who are here today. Also, one of my college classmates and the key contributor to SAGE, Mike Rogowski, who is here, so he's here to answer any questions. So thanks, Mike. But anyway, thank you all for coming today. I'm going to make the transition today from the preclinical to the clinical. When we started and this conference was begun, we actually were focused on the preclinical area, but we have some exciting, what we think, preliminary clinical data that I'm going to share with you today as I go through what SAGE is doing. So SAGE is focused in the area of status epilepticus, and we have a series of compounds either in the clinic or near to the clinic that we have in development. Our lead compound and all of our compounds are positive allosteric modulators of GABA. They all work at the alpha-1 and the alpha-4 extrasynaptic site, which we think mechanistically differentiates our molecules from other molecules currently available. Sage 547 is an intravenous adjuvant therapy under development for superrefractory status epilepticus. And those are the data that I'll share with you towards the end of this talk. I want to spend a little time, though, talking about some of the other molecules we have in development, and those are 689 and 217. Both of these molecules are fully differentiated new chemical entities that are now in IND-enabling toxicology that will be in the clinic either towards the end of this year or in the beginning of next year. Both are somewhat more active at the alpha-1 and alpha-4 site than 547, but they are also differentiated in other ways. 689 has been designed to be used only acutely over the course of a day, intravenous. 217 is an orally available positive allosteric modulator. It's in animals, suggests that we'll be able to use this once daily as an oral agent in humans. 217 is likely to be moved forward into orphan genetic epilepsies, and we're doing some preclinical data now for that, which I won't share with you today. 689 will be positioned as a treatment for second-line status epilepticus. Most of you are familiar with this type of algorithm, what status looks like and how it's treated. The placement of these drugs, as I'll describe in a minute, 547 is adjuvant intravenous therapy for super-refractory status epilepticus. We intend 689 to be an adjuvant therapy for patients as they enter the emergency room in status to be used as an adjuvant in conjunction with second-line anti-epileptic agents. You'll see some of the numbers here, and just as a simple challenge, when you start looking at a disease or disorder where there's no approved therapy, actual demographic data, and as we say in the trade, commercial data is sparse. So over the last year, we've conducted a number of studies with PPOs in the US to establish these numbers, and we're pretty confident that these numbers will stand the test of time, that there are about 35,000 patients per year who will be admitted into the ICU with refractory status epilepticus. There are a lot of challenges to doing clinical trials, and most of you are aware of them, especially when you have a disorder where there's no standardized approach to therapy. Every site has a little bit of their own kitchen chemistry, with all due respect to all the thought leaders who might be in the audience. Everyone has a little different way that they treat this disorder. You have the issue of emergency use and identifying sites where you have adequate patient flow, and also where your actual trial can be conducted. We view these challenges as opportunities, so we've introduced our products for adjuvant therapy to be used in conjunction with standardized care. We do have a compassionate use program in place, and I'll share some of the data we have from that program. In addition, we've spent a lot of time studying the market and the way this disorder is treated, so we've developed internalized algorithms which are proprietary in how we approach sites and how we've been designing our clinical trials. Most of you are familiar with extrasynaptic GABA receptors, and I won't bore you with the details. Suffice it to say that most of us believe there are a number of GABA receptors. Most products like benzodiazepines work at the alpha-4. Our products are both active at alpha-1 and alpha-4. And again, I won't bore you with all the details, but one of the things that we believe differentiates our approach is that during seizures, the synaptic receptor is internalized. And therefore, for therapeutic intervention, only the alpha-4 is available for GABAergic intervention. Our products target the alpha-4, which we think gives us a differentiated approach. You can confirm this more robustly in animals. In an animal model of resistant seizure, you can see on the left, if you induce seizures and harvest the brain, you'll see reduction of synaptic receptors and pretty much stability of the extrasynaptic receptor. Physiologically, that's manifested as the tolerance developing to diazepam in chronic therapy without tolerance developing to sage 547 and related compounds. We've been able to demonstrate this in other preclinical models for both of our compounds 689 and 217, which are active in pharmacoresistant models of status epilepticus, where diazepam is ineffective. We also see good dose response. These are well-behaved molecules, good PK/PD, good predictability, good plasma-to-brain ratios in animals. I want to turn now to the clinical data. While we were filing our IND for sage 547, you all know this is a life-threatening disorder, so we had 4 requests for compassionate use. This slide shows you what these data look like. You can read the age and demographics pretty clearly. The important points to make, all of these were patients in exigent circumstances. All had been in the ICU for weeks to months. All had failed multiple attempts at weaning. What's important is all of these had been are healthy patients prior to their first admission to the hospital, so-called NORSE patients. All of them responded well to 547. All of them have been discharged and cognitively intact, and none of them had drug-related serious adverse events. As a result, we began our phase 1, phase 2 program, which is underway now. You can see the design of the trial. Again, it's based on standardized care with a wean of anesthesia and co-administration of 547, looking for electrophysiologic clearance, as well as the ability to get off general anesthesia. One important point. Our goal in a phase 1-2 methodology study is to look at pharmacologic activity. So we opened the gates to patients we were allowed to be treating. Basically, we wanted to see how this drug behaved across the spectrum of status epilepticus, so we are allowing patients to be enrolled despite their underlying condition, with the caveat that there needs to be an absence of any substantial underlying CNS structural damage. Patients with large space-occupying lesions, large hemorrhagic stroke, and so forth would not be included, but otherwise we allowed enrollment for any other patients. So, sorry, wrong way. Last slide before lunch, if I can figure out how to do this. It's not going the right way. Someone help me. Thank you. Whoever did that, thank you. I may have to hit the bar sooner than I expected. So these are the first 3 patients that we enrolled. And again, the early data with the usual caveats. And you can see there are some differences already. One is that these are relatively new onset cases, although they all failed weaning attempts. And you can see they all met the primary endpoint of the key efficacy endpoint, which was resolution of status. There are some important caveats here that I want to make clear. Only patient 1 is basically a Norse patient. This patient had a subdural, was treated successfully, and was discharged. Patients 2 and 3 had severe underlying disorders. Nonetheless, we agreed to treat them. Both of them responded and met the primary endpoint. However, shortly after treatment was stopped, they then had resumption of some seizure activity. So that's, from a regulatory standpoint, that's a positive challenge and is probative data that you have pharmacologic activity. However, it's unfortunate for the patients. As a result of this, we are amending our protocol. We're looking at higher doses and longer duration, and that amendment has now been submitted to the FDA. But however, we're very excited about these early data. The trial is enrolling. We have about 20 sites opening or about to open, and I look forward to sharing more data with you in the future. So with that, let me stop and see if I have any time for questions.
Thank you very much. So what's the FDA going to, this is a tough area to study. What's the FDA going to require of you to get this drug on the market? Let's assume that that things go well. What are they going to require for you to get this on the market?
You know, of course, I know exactly what the FDA will say. No, I think this is being designed as an intervention trial, and we've approached the agency with that in mind. So we believe we can attain approval with an acute endpoint at this point as an intervention, much as you see with antipyretics or, for example, pressor agents, despite the underlying etiology. So we think that will be a suitable label, and indeed, that's how we've been approaching it.
Are you going to need more than one trial?
A lot of that will depend on what the nature of the activity is.
All right, that's enough. Okay.